Hello dear viewer, and a warm welcome to this web lecture on benefit risk assessment. My name is Ola Caster and I'm a senior researcher here at the Uppsala Monitoring Center with several years experience from research in the area of benefit risk assessment. In this 1-hour lecture I will go through in the first half the basic concepts of benefit risk assessment and try to make you understand why it's such an important but also such a complex area. And in the second half I will go through some of the methods that have been recently proposed to support, for example, us pharmacovigilance professionals in this very crucial task.
Some of the slides that I will show you contain references that will be displayed at the end of the lecture. If you have any questions on these or on anything else related to the content, feel free to contact me at the email address shown here. So let me start by saying a few words about terminology. Terms like benefit, risk, harm, effectiveness, etc. mean different things to different people. The European Medicines Agency, the EMA, performed a study among domain experts in different national regulatory agencies in Europe, and even within that group the differences were remarkably large. And the specific construction benefit risk assessment is admittedly a bit strange because benefit is often considered something good that is manifest and certain, whereas risk implies the potential for something bad with some likelihood. However, this specific term, benefit risk, is the one that's most often used, and so for simplicity I will follow that convention. In any case, I believe it's more important what one means than what specific term one uses, and so I will start to try to explain what I mean by benefit risk assessment. So here is my own tentative definition, which is that benefit risk assessment aims to answer either or both of the following questions. First, how useful is a medicine at a certain dose in a given indication for use in a given patient population? Or how useful is a medicine at a certain dose in a given indication for use in a specific individual patient? Now, this can apply both pre- and post-marketing, although the focus in pharmacovigilance is obviously post-marketing, that is, after a medicine has been allowed to enter the market. Both the population perspective, which is captured in this first question, and the individual patient perspective, which is captured in this second question down here, are important. Although the focus of this specific lecture will be the population perspective, which is why this first question here is given in bold. Now, this definition that I've given here is based on what kind of answer a benefit risk assessment will give, rather than how it is performed. Obviously, this answer will depend on the benefits and the risks, which is why this definition hopefully makes some sense. A very relevant question is why benefit risk assessment is important. Most often it's used to support some kind of decision making. And at the population level, as you see up here, those decisions typically concern licensing, that is, whether a medicine should be allowed to enter or remain on the market, or policy recommendations. At the individual patient level, clinical treatment decisions are the most relevant. All of these decisions concern the availability or the use of medicines, and both the diseases for which those medicines are used and the potential adverse effects they cause greatly affect morbidity and possibly mortality of lots of patients, as well as the quality of life of those patients. And that is why I think benefit risk assessment is so important. It's ultimately about the life of a lot of patients. Now, one aspect that is easily overlooked is that benefit risk assessment is not a static thing. It's a very dynamic process. And at each point in time, the assessment is conditional on the currently available information. So this sketchy figure here shows you just a few examples of how our understanding of a medicine's usefulness can change over time. And in pharmacovigilance, we are often concerned with detecting previously unknown adverse effects, which I've tried to indicate here in the colored box. When this happens, the overall benefit-risk balance of the medicine will go down inevitably. It's difficult to say exactly how much it will go down, but it should go down. But it can also go the other way. For example, we can learn that the medicine is actually more effective than we previously thought. If this happens, its benefit-risk balance will go up. So here is one example where we have identified new data from the post-marketing phase that indicates a higher effectiveness than we previously thought. There are lots of things that happen. A third example that I've given here is that a superior alternative medicine is marketed, in which case the relative usefulness of the medicine that we're looking at will drop dramatically. So that's indicated here. And these are just 3 examples. Lots of things can happen over time, but the important message is that it is not static, it is dynamic. Now, I defined benefit-risk assessment as answering questions about the usefulness of medicines. This begs the question, what is a useful medicine? So I've tried to put down some kind of best-case scenario that I believe and hope most of you would agree with. And what I've written here regarding the medicine's beneficial effect is the following. The medicine induces a clear and lasting improvement on the course of the disease, or alternatively completely cures or prevents it, for a high proportion of treated patients. And for the adverse effect, I've written the medicine has few adverse effects, all of which are trivial and transient in nature, and all of which occur in a tiny proportion of treated patients. The problem, of course, is that this is not a realistic scenario in practice. So instead, I will try to highlight specific aspects of this description that can help us, hopefully, to evaluate the usefulness and hence the benefit risk. So the first aspect that I've highlighted is frequency. And it is quite clear that whatever good the medicine is doing, the more patients that experience that good, the better. So in this example, I've written that a high proportion of treated patients experienced the benefit. And conversely, obviously, for the adverse effect, for any adverse effect, the fewer patients that are affected, the better. And in this example, this best case scenario I've written, it was a tiny proportion for all of the adverse effects. So the second property is seriousness. And for adverse effects, we are very used to considering seriousness, at least in pharmacovigilance, and it is apparent that the more serious the effect is, the worse it is. And in this made-up scenario, I stated that all the adverse effects were trivial in nature, for example. But we can apply a similar reasoning for the beneficial effect. And here the question is, how serious is the disease that we're treating or trying to prevent? And to what extent is the level of seriousness lowered by the medicine? So in this case, it is a bit of a relative thing, but it is possible to apply the seriousness concept also on the beneficial effect. The third and final property, or dimension as we call it as well, is duration. And obviously for a beneficial effect, we want it to last for as long as possible, ideally indefinitely, as would be the case, for example, with a complete cure. And for the adverse effects, again, it's the opposite that we're looking for. A transient effect has less negative impact than a persistent one that has otherwise similar attributes. And at the extreme end, obviously, are irreversible adverse effects, of which the lethal ones are an important and especially troublesome subset. But in this specific made-up best case scenario, I've written that all of the adverse effects were transient. So, in summary, what I've tried to do is to identify these three concepts or dimensions, frequency, seriousness, duration, as the three key dimensions to consider benefit-risk assessment. But if we're talking about an individual patient, as indicated here, it does make more sense to substitute frequency by probability. Obviously, these two concepts are tightly connected. Now, these concepts that I've presented here are not new in any way. As you see, there's a reference to a paper from 1996 here. But I do think they offer a very good summary of what is important to consider to be able to properly evaluate the medicine's usefulness and hence make a benefit-risk assessment. I cannot claim, obviously, that these three dimensions perfectly capture everything. However, I am confident that if they are well characterized for all relevant effects, then one has a very solid foundation on which to base a benefit-risk assessment. I think two things are especially worth emphasizing here. First, these dimensions have to be assessed for each effect that we choose to consider. Otherwise, it doesn't really make any sense. Otherwise, it's not complete. But how we do this, for example, what kind of data that we look at, can be very different depending on what kind of effect it is. And secondly, as I said earlier, it may appear strange or awkward to consider seriousness for the beneficial effect or effects if there are several. But it does make sense, I think, if we consider how the seriousness is altered, changed relative to the natural course of the disease. So what I will do now is I'll simplify this slightly, at least on the surface, and I will replace seriousness and duration with a single concept that I call desirability. And by that I mean how desirable, or in some cases undesirable, is a certain effect, and that will depend both on its seriousness and on its duration. This may feel awkward or strange at first, but if you think about it, it's really not that strange. And the reason that I do this aggregation of these two is because many methods that are used to proposed in benefit-risk assessment are based on two rather than three dimensions, and we'll get to this later and look at it in more detail. It is very important, I think, to realize that frequency and desirability, as I've now introduced, are inherently different in nature. And the overview that I've given you here lists some of the key differences. Most obviously, I think, frequency is an objective thing, whereas desirability, especially the seriousness component of it, is subjective. For example, if we study a certain patient population and find that 50% experience a specific beneficial effect, it doesn't really make any sense to argue about that frequency. We can argue about the way it was estimated, but the frequency, basically, there is a frequency and it is what it is, basically. But desirability, on the other hand, is clearly subjective, since it depends on how good or bad we think that a certain effect is. And there is really no obvious right or wrong in this. It essentially comes down to preferences and value judgments. So that's the first point here. The second point is that frequency is relatively straightforward to measure, and it has a natural scale. It's a natural interval scale. And by that I mean that it is twice as bad if some adverse effect, for example renal failure, occurs in 2% of patients compared to occurring in 1% of patients. It is that simple. Twice as common, twice as bad in the case of an adverse effect. But desirability, at least the seriousness part of it, doesn't have a natural scale, and it's also very difficult to measure. And partly because of how easy they are to measure, but also because I think of what has traditionally been considered important to measure. Frequency is nearly always measured in situations where that is feasible, for example in clinical trials. Desirability, on the other hand, is rarely measured, even though I think that this might be gradually changing with time. On the whole, I would say that frequency is a very well-established concept in pharmacovigilance and pharmacoepidemiology, whereas desirability is more of an alien concept. Although, again, this might be slightly changing with time, hopefully. One point that I would like to make here is that if we really want to make progress in the area of benefit-risk assessment, we must acknowledge that there are subjective components of this, and we must learn to handle them. Next, I would like to try to show you that even if we're able to capture all of these dimensions, it is far from easy to perform a benefit-risk assessment. So what I've sketched here is a fictional example, it's all made up, of a medicine used to manage moderate to severe relapsing remitting multiple sclerosis, or RRMS. So there is a lot of information here on this slide, but basically what you see in the upper part here is information on the beneficial effect, and down here is information on 3 specific adverse effects that we're considering. And frequencies that apply in patients treated with the medicine are given in yellow, as here, and frequencies for the non-treated patients are given in purple, here. So what we do see, first of all, is that the medicine has a positive effect. Half of the treated patients get better, 1/4 experience no change, and 1/4 get worse. I've also tried to define what better and worse mean in this case. So either the so-called EDSS, which is a measure of disability in multiple sclerosis patients, goes down from about 4.5 to 3 or below, or else it goes up to 5 or above in case the patient is considered to get worse. And these effects are assumed to be persistent for 1 year, as I've written here. The first, and in contrast to what happens with those that are treated, as you see, among the non-treated patients, they fare considerably worse. None of them get better, half of them see no change, and half of them actually get worse. So if we then go down and look at the adverse effect, the first one is new onset diabetes, which is also considered to be persistent for 1 year. and 1% of the treated patients experienced this compared to 0.1 for the non-treated ones. The second adverse effect is transient paresthesia for 1 to 2 months, which occurs in 2.5% of the treated patients and only 0.2% of the non-treated patients. And then finally we have myocardial infarction down here, 0.1% compared to 0.1%. So this is a simple, in quotation mark, example with only 3 adverse effects that are very well described here, and also all the frequencies are assumed to be known perfectly. But still, it isn't really that simple, is it? I mean, is this a useful medicine? Should it be allowed to be used? Should it even be recommended for use? We have all the information there, but a whole lot of processing has to be made to be able to reach a conclusion, to be able to answer those questions. And specifically what we need to do is we need to assign in some way desirability to all of the different effects that we are considering here, meaning multiple sclerosis in just both the low, the middle and the high range EDSS, for persistent diabetes, for the paresthesia, and also for the myocardial infarction. And then we have to somehow combine that with the information we have about the frequencies, and finally aggregate everything together. And of course here, assign desirability, that doesn't necessarily mean in a that we have to do it in a quantitative way, but somehow we need to take into account what the level of desirability is, or else we will basically imply that the desirability level is equal for all the effects. Now, one thing we know is that no matter how complex this really is, this is something that is done in practice all the time and everywhere, because decisions that hinge on this kind of information that I've just presented here have to be made, and there is simply no way around that. Often, however, we just don't really know how these assessments are made. And I would say that we cannot realistically expect that humans will consistently make sound decisions when trying to process this wealth of information without any kind of support. Yet, that is basically what we're supposed to do. And then, of course, I have to emphasize this again. The specific example that I showed you here is very, very simplistic compared to the reality. And just to further try to get that complexity message across, I will point out 2 specific complications that were not in that first slide, but that are nearly always present in real life. The first one is comparativeness. It is very rare that there are no alternative treatments available. And here I've added corresponding information to the one you saw previously, but also for an alternative medicine. And those frequencies are written in black here in this middle column. So we see that this existing medicine is slightly less effective. It's only 40% that get better compared to the 50% for the new medicine. At the same time, it is safer when we consider the 3 adverse effects from before. As you see, this alternative medicine has no elevated risk here compared to the non-treated patients. However, this alternative medicine has a slightly increased of another adverse effect, lethal infection, compared to the non-treated patients, which the new medicine doesn't have. So that further complicates things. And now, what do we do if we want to get to the point where we can actually recommend either of these 2, if any of them? If we think about a typical pharmacovigilance scenario, it may well be that there is also a time difference between these pieces of information, as I talked about earlier. We might, for example, know from the time of marketing about these two effects, the diabetes and the paresthesia, and we have made the comparison to the alternative, and we have considered that, well, this new one has an acceptable benefit-risk profile. But then, sometime later, there is an emerging signal of this myocardial infarction, which may or may not put everything upside down. At least it calls for a reassessment. So that's another thing to consider here. The other complication, which is a very significant one, is that information is never entirely certain. And in fact, uncertainty is often quite considerable. So I've tried to indicate this here by replacing the point frequencies that we had before by intervals of possible frequencies, as you see here. And of course, in the pharmacovigilance setting, it's also relatively common that we are not even certain that there is a causal link for one or more of the adverse effects. That is, we're not even certain that there is an increased risk. And this is the typical situation for emerging signals, as I talked about for myocardial infarction earlier. I have now covered a lot of the basics, and I have tried to emphasize the complexity and the challenges. So I think it's now time to turn the corner, so to speak, and instead talk about possible solutions. But before I do that, I'd just like to summarize the first part of the lecture, the things that we have covered so far. And the first point here is that benefit risk assessment is important, which I hope you will agree with. And the main reason is that important decisions about the use or the availability of medicines have to rely on benefit risk assessments, whether they are explicit or not. And those decisions will affect patients' morbidity, mortality, and quality of life. The second point is that frequency, seriousness, and duration are three key dimensions that together capture most of the essential information. And sometimes it makes more sense to combine seriousness and duration, and I've chosen to call that desirability. Third, and finally, the sheer amount of information, as well as its complex nature, makes benefit risk assessment a very challenging task, and it really isn't suitable to do manually. If one accepts these premises, it actually follows logically, I'd say, that the ethical thing to do is to look for methods that can support us humans in the benefit risk assessment process. Because if we do not, then we run a clear risk of making decisions that are not sound, and that in turn will cause unnecessary morbidity, mortality, or decreased quality of life. And the key word here is support and not replace. So what I'm not suggesting that we simply hand over decisions to algorithms or some other kind of artificial intelligence. I'm merely saying that it would be good to have solid supportive methods and tools. And this is an area that I have pursued personally. For example, on the screen here you see the front cover of my PhD thesis that deals with these kinds of things. But there has been an interest in research on such methods for a long time now. And I'd like to mention the CIOMS-4 report from 1998 as an early milestone. But since then the area has accelerated, and especially I'd say in the last decade or so. So from here on I will talk a lot about formal methods, as I've chosen to call it. And so this is a broad term, but basically I mean just some kind of pre-specified algorithm or procedure to collect analyze and combine data and other relevant information. And it does not have to be quantitative simply because it is formal. I've here listed 3 potential benefits that are reasonable to expect if using sound formal methods to support benefit risk assessment. One is increased transparency, because it will become, if you use this, it will become inevitable to document well the underpinnings of a decision. And the next one is increased consistency, which follows from repeatedly using the same pre-specified method. So it's more likely that different or the same assessors will reach the same decision given the same data and the same value judgments. The third one is increased robustness, because there should be a lower risk to exclude crucial elements. And that is a risk if you're relying on human introspection alone. And also because a formal method is explicit, it will make clear to us what knowledge gaps are present. I should mention that these 3 statements here are more arguments at this point than they are facts. And as far as I know, no one has empirically demonstrated that they are true. But they are, I would say, very reasonable. For your reference, I've here listed several properties that I think formal methods for benefit risk assessment should possess. And some of these should be hopefully evident from the discussion we've had so far. For example, the ability to accommodate both frequency and desirability, to allow for multiple effects, and basically all effects that are relevant, to allow for comparisons between medicines, and also to handle uncertainty in some way, as written here. I haven't talked a lot about data sources so far, but this is a key point, obviously. And specifically, the data that is relevant to use is often very different for different effects. And for example, beneficial effects are often studied extensively in clinical trials, and sometimes also in observational studies on using real-world data. Adverse effects, on the other hand, especially the rare ones, for those who may only have data from spontaneous reports, and so on. So the ability to accommodate disparate data sources is also a key property. Further down we have transparency, meaning that it is reasonably easy to see what the method is doing. We have soundness, meaning that the method will produce results that are coherent with the inputs. And finally, practicality means that the method is actually a feasible tool for the intended user. I will very shortly move on to show you actual methods and real examples where they have been used, but I would first just like to reiterate that these methods need not be either quantitative or prescriptive, which may be a common misconception. And in this context here, prescriptive simply means that the method would produce a result that recommends a specific decision. Method can also be just simply descriptive, and we'll get to that shortly. Now, this is a table that I will obviously not present in detail. I've included here several examples of methods that have been proposed over the last decade or so. And I recommend to consult these references if you want to learn more, both about the approaches that I will cover here and those that I will not cover. I should add that this development has been mainly an academic endeavor so far, which has to some extent created a slightly chaotic situation. And there are even lots of other methods that are not even mentioned in this picture here. However, I should also say that regulators and industry have been more and more involved, especially of late. what I will do is I will present in some detail two specific group of methods that have been identified in several reviews as promising, and that also appear to be the ones that are most used so far. And this applies in particular to so-called multi-criteria decision analysis, or MCDA as it's often abbreviated. And MCDA belongs to one group of methods that I here call criteria-based. As you see in this slide, there are lots of abbreviated names like PROACT, URL, BRAT, and so on that you probably don't need to remember. But the purpose of putting this table here is to try to help you get a feeling for the different steps that make up this approach. And this follows a more or less linear structure. So it starts out by defining the nature and the context of the assessment, and that can be, for example, questions like what medicine are we looking at, what indication, what population, what dose, what are the comparators, for whom is the assessment made, and so on. And then comes the step of defining the objectives, and that includes questions like why is the assessment performed? In other words, what kind of decision is to be made? Is it a licensing decision? Is it a clinical decision? Etc. Then the third step is to identify alternatives, and that's simply to answer the question which options are available in this decision. Then we have the step of selecting key outcomes, which are here called criteria, and then to create a value tree, which is basically your model that you're using. And I should mention here that the criteria correspond effectively to what I have so far called effects. So it's just a slightly different terminology. Then in the next step, one is supposed to select data sources and extract relevant data. Further, to fine-tune the criteria definitions and the model structure. And then to rank or weigh the criteria by importance. And this is where the desirability dimension is being considered. Then we have finally the step where we are supposed to display and interpret all the information. And for MCDA, additionally, we perform a quantitative aggregation. So it's really only in this final step here that quantitative methods like MCDA differentiate themselves from other more descriptive methods. And in MCDA, as we will see shortly, each option is actually assessed quantitatively based on the importance that is given to each criterion, and also a quantitative measure of how good each option is with respect to each criterion. And whether they are good or bad is usually based on frequency information. So the example that I will use to illustrate this approach is natalizumab, which is an immunomodulating monoclonal antibody that was first approved in 2004 for relapsing-remitting multiple sclerosis. Sometime after its approval, there was a controversy, since it was found at that point that natalizumab actually increases the risk for something called progressive multifocal leukoencephalopathy, or simply PML. And natalizumab was actually allowed to stay on the market despite of this finding. But it is interesting to compare to another monoclonal antibody, ifalizumab, that also operates in a similar way. It decreases the immune response, and that was also linked to PML. But in contrast to natalizumab, ifalizumab was in fact withdrawn from the market. And it is reasonable to assume that these very different decisions were at least partly related to the difference in indications, because natalizumab was used in psoriasis, which is obviously a very inconvenient condition, but it's still clearly less serious than multiple sclerosis. So what I'm presenting here is a retrospective analysis performed by Nixon and colleagues, where they use MCDA as a support to assess natalizumab in the light of the PML discovery. So this analysis was performed several years after the actual decision. It was more an analysis to showcase the method and how it works and what it can do and so on. And what you see here in this table is the context and the objective of the assessment. And the important thing here is that it's performed from the perspective of the regulator, which is stated out here. And the decision that is supposed to be made is whether natalizumab should be withdrawn or not. That is stated up here in the objective. So this is the value tree that they used, and such a tree makes it very easy for us to see what specific effects or criteria that were considered. And you should note specifically here how the beneficial effects and the adverse effects are modeled in separate branches of the tree, so up here and down here, and then that they have further subdivisions underneath. And the view here is zoomed in. It's the same picture, just zoomed in a bit more to specifically highlight PML here in the value tree. And now, if this were a descriptive criteria-based analysis, what one would do is one would present here the available data for the various alternatives on all of these criteria that are considered, and the relative importance of the criteria would be assessed qualitatively in some way. And in this case, in the case of a descriptive model or descriptive analysis, this model would be used as a tool more or less to support the assessor, to highlight, to show what are the criteria that are being considered, help you qualitatively assess their relative importance, present the available data for the different options on these criteria. So it would be a tool to support the assessor, but it would not actually prescribe any particular decision option as the best one. But in MCDA, which, as I said, is a quantitative and prescriptive method, we take this one step further. So here the different criteria are assigned numerical weights. So here you see the different criteria, and here you see the weights that are given to them. And as I said, the higher the weight, the more important it's considered to be. And in this example, we see that PML here has clearly the highest weight of all the criteria, which is because it is such a serious adverse effect. But it has a very small effect actually on the overall benefit risk. So this negative number here, minus 0.8, is the effect, the overall effect on PML on the benefit risk. And you see the benefits up here have clearly more clearly higher positive contribution compared to this negative contribution. And overall, this is the benefit risk is considered to be very positive for natalizumab compared to placebo, which is indicated by this positive number down here. And I should also add the other options that you might have noticed in the previous slide, the alternative medicines are not shown in this particular figure. But ultimately, in this case, the retrospective result of the assessment is coherent with the decision that was actually made. Yes, so I have now covered the basics of the criteria-based methods. We have looked particularly at MCDA, in this case of natalizumab. So next I will briefly introduce the fundamentals of the decision tree-based methods by looking at another classic example from reality. But first I'll introduce what these decision trees are about. And decision trees are also, like the criteria-based methods, rooted in decision analysis. A key difference here is that these decision trees always take the perspective of an individual patient, even if the assessment is for a specific population. And what a decision tree does is it depicts possible lines of events that we call branches, whose probability and utility are used to compare the usefulness of the available options. And the underlying theoretical framework here is quite complex, but a reasonable simplification is to say that the probabilities of these events correspond to the frequency dimension, and the utilities correspond to the desirability dimension that I have introduced earlier. It should also be mentioned that decision trees are by nature both quantitative and prescriptive, just like MCDA. They will suggest one alternative as the most useful one, based on which alternative that has the highest so-called expected utility. And the example that I will use here is the medicine alosetron. And it has quite an interesting history, because it was approved first in the year 2000 for irritable bowel syndrome in women, and then it was taken off the market later the same year because there were concerns of serious gastrointestinal complications, and a few of those were even lethal. Then there was considerable debate and reassessment, and finally alosetron was reinstated on the market in 2002. Another interesting aspect is that alosetron is only available in the US still. And I think all of these different points have made alosetron a very popular example on which to apply various benefit risk assessment methods, because it isn't entirely clear here what is the right call, so to speak. And the issue is further complicated by the fact that alosetron is not terribly effective compared to conventional treatments. And roughly speaking, the clinical trials suggested that a little over 40% of the patients on alosetron got well, compared to a little less than 30% of those on conventional treatment. So it's not a huge difference. One author summarized this issue like this: the case of alosetron illustrates a challenging dilemma. When can the possibility of symptomatic improvement in a condition that is not life-threatening justify the risk of serious complications, including death? So here is one decision tree that has been used in the assessment of alosetron. This zoomed-in view here should make it a bit easier for you to distinguish the individual elements of the tree. So the green square to the very left here shows which alternatives that are considered, and in this example it's alosetron and standard care. The red circles that you see here are so-called event nodes, which outline what possible events are considered. For example, if we look at what can happen following complication, we consider 4 different events. First, that it is a non-serious complication that can be managed in outpatient care, that it requires hospitalization, that it requires surgery even, or in the worst case, that it's lethal. And this approach here of using decision trees requires that probabilities are assigned to each possible event that is considered. So we would have to put probabilities on these different considered events here, for example. And also that we, in the end, then compute a total probability for each branch, which we do by multiplying the individual probabilities here to get something for an entire branch. The blue triangles that you see here and also here are so-called consequence nodes, and there is one consequence node for each branch in the tree, and each such node is assigned a utility, and the alternative, so in this case alosetron or standard care, the alternative that has this so-called highest so-called expected utility, which is the probability weighted sum of the utilities that are considered for that alternative. That option is the one that's considered the preferred one. And as I mentioned earlier, this makes this approach inherently prescriptive. And in this particular example with alosetron in this model, without going into any kinds of details, the conclusion was that the overall benefit-risk balance was more positive for alosetron than for standard care. And subsequent analysis with other methods have reached the same conclusion. So we're getting near the end of this lecture. I will soon get to a summary, but first I'd like to offer you some final thoughts on the area of formal methods to support benefit-risk assessment. And my first point here is that this is still very much work in progress. It's an active area of research, and we're still quite far from established practice. What I have been able to cover here in this lecture is really just scratching the surface. However, I do foresee that progress will continue in this direction, partly because I see interest both from academia, regulators, and industry, and partly because there are major opportunities that would be beneficial to everyone, and not least patients. And I have listed 2 important opportunities here. First, the potential to more consistently make sound and well-informed decisions, and also an increased openness, which should hopefully increase public trust. Having said this, there are clearly some significant challenges that have yet to be properly addressed. And first of all, devising methods is one thing, but it is painfully clear that they cannot function well unless we have the required data. And this becomes even more challenging the more we try to make increasingly more stratified or personalized assessments. So far, clinical trial data is dominating, which is relevant for initial licensing, but not so much for subsequent assessment. I think more real-world data are required for all kinds of patients that are using a specific medicine. Also, because benefit-risk assessments have a subjective component, it becomes difficult to develop and not least evaluate proposed methods. Because if there is no objectively right or wrong answer, then how do we know what method is working well? So I think other criteria are needed here, and as a field, I think we're far from from cracking this nut at the moment. I put down here my very long-term vision for this field, which is that human experts use sound benefit-risk assessment methods to support all important decisions related to medicines, including development, initial licensing, subsidization, surveillance, that is in what we're doing in pharmacovigilance, and also individual therapy. And clearly we are far from this long-term vision at the moment, and it is very difficult to speculate when or even if this will become reality. So then, to summarize, in this lecture I've tried to explain and motivate why benefit risk is so important. These assessments affect decisions that in turn affect patients' morbidity, mortality and quality of life. At the same time, they are very challenging, primarily due to the wealth and the diversity of the information that has to be processed. Secondly, I've briefly touched upon the dynamics of benefit risk assessments. They are to some extent perishable, and they need to be updated as new important information comes in. Further, I have introduced the key concepts of frequency, seriousness and duration, and I think it's very important to realize that these are different in nature, especially if we look on the objectivity-subjectivity scale. And then in the second part of the lecture I introduced some formal methods that have been generally considered promising as tools to support humans in the benefit risk assessment process. Criteria-based methods, and especially multi-criteria decision analysis, or MCDA, is one group of methods that is worthwhile to remember, and decision tree-based methods is another one. And finally, the two examples of natalizumab and alosetron are interesting and I think useful to keep at the back of one's mind when browsing through the literature on benefit risk assessment. Well then, that was all I had to say today. Thanks very much for listening, and I sincerely hope this was useful to you. And any feedback is much appreciated, preferably via email using the address shown here at the bottom. I will now just quickly flip through 3 pages of references. The first one is for those that were used in specific slides, and then there are 2 that cover the long list of examples of methods that I showed earlier. So if you have a particular interest in these, please just pause the recording when appropriate and research those further. Thanks again very much for listening, and goodbye.